At Moderna, we believe messenger RNA represents an unprecedented opportunity to develop medicines for patients, one that could have a dramatic impact on the course of human health and disease. In order to understand why we believe that, we have to pull back and ask a very fundamental question, which is, what is life and how might messenger RNA impact it? Now, life is really just the flow of information in a biological system. The ebbs and flows of that information dictate health and disease. And that's true in every cell in our body, throughout our bodies, and throughout all forms of life. In order to explore that more deeply, we must follow the flow of information in cells. And in cells, information is stored in the DNA. A DNA is a double-stranded helix of tightly wound genetic information that is itself wound around a set of proteins called histones that are themselves then tightly wound around each other into chromosomes. It's analogous to those old tape drives in IBM computers where, in order to access that information, what cells have to do is they have to go find that right tape drive, unwind it, and start to make a working copy. That process of unwinding and making a working copy is called transcription, where a series of proteins will search that DNA, unwind it a bit, bind to the DNA, and start a process of making a pre-messenger RNA molecule. That pre-messenger RNA molecule, when done, will go off and be spliced by a whole different group of proteins called the spliceosome that will edit out certain pieces, leading to a messenger RNA. That messenger RNA is the first fully intact copy, working copy of the information of life that then leaves the nucleus and goes and does all of the work of life. It does that by seeking out ribosomes in the cytosol. These ribosomes translate the messenger RNA into all of the proteins of life, including secreted proteins, transmembrane proteins, mitochondrial proteins, and even proteins that go back into the nucleus and do all the work of storing that information or selecting information to be unwound to make more messenger RNA. So messenger RNA really is the central molecule in all of biology's processes. We call it the software of life. Now, given that power, it's reasonable to ask whether others have pursued it as a therapeutic approach. And in fact, many of the more exciting technology platforms in biotechnology today are. One example is siRNA. siRNA specifically binds to and degrades messenger RNAs inside of cells. It selects a specific messenger RNA relative to others, thereby changing the flow of information. Another example is gene therapy. In gene therapy, DNA is introduced from the outside and translocates into the nucleus of cells, where either episomal DNA or DNA that is inserted into the genome of the host will go through those same processes of transcription into pre-mRNAs and splicing into final mRNAs that then get exported out of the cell. In many senses, gene therapy is an indirect way to increase or add a specific type of messenger RNA to cells from the outside. So all of these approaches are about modifying the types or amounts of messenger RNA inside of cells so that you can have a phenotypic effect in a disease. Our approach has been more direct. Can we put messenger RNA directly into cells to create proteins, both endogenous proteins and foreign proteins? And if we could, we believe that messenger RNA could be used to make the full set of proteins in life.